Hello everyone, hello students. Today we are going to discuss about the brief notes on cell cycle. Have you ever wondered how a multicellular organism will produce from a single cell zygote? They produces a multicellular organism by the process of cell division. Before they enters into the cell division, they passes through a sequence of event or series of event. That series of event is known as cell cycle. The cell division also helps in gamete production, wound healing, and in case of unicellular organism, they helps in reproduction. Each individual required a certain time for their cell division. That time is known as generation time. Each individual have different types of generation time. Like human being have 24-hour generation time, and in yeast that have 20-minute generation time. The cell cycle is divided into two phases, interphase and mitotic phase. The interphase is also known as preparatory phase. As before the cell enters into the cell division, they prepare itself for cell division. They prepare themselves. They prepare the cellular organelle. They prepare the centriole, which are play important role in cell division. The interphase is divided into four phases, that is G1 phase, G0 phase, S phase, and G2 phase. Again, mitotic phase is divided into karyokinesis and cytokinesis. Further, karyokinesis is divided into prophase, metaphase, anaphase, and telophase. So let's start from the G1 phase. G1 phase is the first gap phase or first growth phase of the cell cycle. It is also known as post-mitotic phase, as this phase occurs just after the mitotic phase. That's why this is known as post-mitotic phase. Cells releasing from the M phase enters into the G1 phase. That's why this is known as post-mitotic phase. In G1 phase, cellular contents doubles their number, as well as mRNA synthesis or transcription and protein synthesis or translation occurs in G1 phase. After the cellular organelle and other materials of cell enters into the S phase, the second phase of cell cycle. This is also known as synthesis phase. In synthesis phase, the DNA replication or DNA synthesis occurs. That's why this phase is also known as S phase. That double DNA and other cellular organelle passes to G2 phase. G2 phase is also known as growth 2 phase or gap 2 phase. It is also known as pre-mitotic phase, as it occurs just before the mitotic division. That's why it is also known as pre-mitotic phase. In G2 phase, cell grows and prepare itself for mitosis. There are some checkpoints. The main checkpoint, G2 checkpoint, which are discussed in later. That checkpoints play important role when they passes from one phase to another phase. G2 checkpoint play important role when the cell enter from G2 to M phase. M phase is divided into karyokinesis and cytokinesis. That karyokinesis is nuclear division. Karyokinesis comes from a Greek word, that is karyon. Karyon means nucleus. Nuclear division is known as karyokinesis. Karyokinesis can be divided into four types: prophase, metaphase, anaphase, and telophase. Then that cell divided by cytoplasmically. That means that in cytokinesis, the cytoplasm division occurs. Cytoplasm divides. In animal cell, cytoplasm divides cell Furrow method. In plant cell, they divides by cell plate method, and they produces two daughter cell as a result of cell division. So let's come to the control of cell cycle. For the proper functioning of the cell cycle, they should be managed in a sequential manner. Otherwise, the cell would be developed into a tumor cell or cancerous cell. For their control, there are number of checkpoints. there are mainly 4 checkpoints which are involved in the cell cycle. First one is G1 checkpoint, which is present just starting of the S point. Then intra S checkpoint, then G2 checkpoint, then spindle assembly checkpoint. The G2 checkpoint checks the proper folding, proper formation, proper duplication of all the cellular organelle before entering into the cell division. And the spindle assemble checkpoint checks the proper attachment of the spindle fiber with the kinetochore of the chromosome, which are present just behind the centromere. After the proper attachment, they separate easily. If there are they cannot be attached properly, they would lead to different type of ploidy condition or sex chromosomal aberration condition or autosomal aberration condition. Then comes to the regulation of cell cycle. The cell cycle can be regulated by a protein, that is cyclin protein. Cyclin protein also control the function of CDK enzyme, that is cyclin dependent kinase enzyme. There are a number of cyclin protein and number of CDK enzymes. Like in G1 phase, the cyclin protein D and CDK protein 4 and 6 are involved. In G0 phase, cyclin protein E and CDK enzyme 2 are involved. In S phase, cyclin protein A and CDK enzyme 2 involved. In G2 phase, the cyclin protein A and CDK protein 1 is involved. In M or mitosis phase, the cyclin protein B and CDK protein 1 is involved. These cyclin and CDK enzymes play important role in the controlling or the regulation of the cell cycle. That's all about the cell cycle. Hope you understand well. So thank you.